This video is Chapter 8 Notes, Part 2, all about checkpoints. In Part 1, we went through the stages of mitosis. To remind you, the purpose of mitosis is to replace cells. So always cells are constantly dying and getting replaced in an organism. For example, in a human, they say that a human gets an entire new set of skin cells every 7 days, which is kind of weird to think about, but you're constantly shedding skin cells that are dead, and your body is replenishing those through the process of mitosis. Also, mitosis is used for repair. So if you get injured, whether it be a paper cut or a more traumatic injury, your cells will repair that injury by replicating, by doing mitosis. Also, asexual reproduction uses mitosis. And then also growth of an organism. So just meaning physically getting bigger in size. The steps of mitosis begin with interphase. During interphase, the cell is predominantly spending its time in G0. It is waiting for some signal to divide. Once it gets that signal to divide, the cell will begin mitosis and begin division. But there's several checkpoints that occur during the steps of mitosis to ensure that everything is going correctly. And if it's not going correctly, the cell will try to repair any errors and correct any errors, or the cell will be forced to kill itself because it identifies that it would not be good for the organism to have replication of these damaged cells. So cell division is controlled by checkpoints. Checkpoints are a huge idea in the AP curriculum when it comes to mitosis. So just memorizing the steps of mitosis will not be sufficient of an understanding of mitosis. Huge is the checkpoints. The checkpoint acts as a system of control for the cell to make sure that it's dividing only when it should, and that it is dividing properly. So to remind you, during interphase, after the cell gets told to divide, the cell will get kicked out of G0. After G0 comes G1. So the first set of checkpoints is the G1 checkpoint. During G1, the cell is going to get physically bigger in size. The cell should only begin this process of starting mitosis if the cell is sure that there's enough space, because obviously replicating and dividing makes more and more cells. So if there's not enough space for those cells, it would be a waste of energy for the cell to divide if that resulting cell is just going to die. And then also, is there enough food? Same concept. What would be the point of spending energy to divide if then there's going to be a shortage of food? So the G1 checkpoint is the first of 3 checkpoints. Is there enough space and enough food? The G1 checkpoint is controlled by one of the most researched proteins when it comes to cancer research, which is a protein named P53. P53 is also called the tumor suppressor protein. A tumor is a clump of cells that continues to divide even though there's no physical space for those cells, and thus tumors tend to grow 3-dimensionally in an area where a mass of cells should not exist. So cancerous cells, tumor-forming cells, tend to ignore the G1 checkpoint. They don't care if there's enough space. They don't care enough if there's enough food. They should not divide, but they continue to divide. So some cancers are caused by a mutation in this protein called P53. P53 should suppress, it should stop a tumor from forming. So P53 is like the little gatekeeper here saying, stop, there's not enough space, there's not enough food. But if somebody has a mutated copy of P53, the cell might bypass that checkpoint, ignoring the fact that there's not enough space and food. Interestingly, a lot of cancer research has been done on elephants because in the animal kingdom, elephants live a long time and they don't really ever get cancer, which is kind of unique compared to humans. You know, cancer is extremely common amongst the human population. So a lot of research has been done with elephants. And what scientists have found is that elephants have 20 copies of that gene. So if 1 or 2 or 3 or 4 or more copies are mutated and screwed up, they still have a protein that is saying stop because they have so many copies of P53 that the protein is still there saying stop, do not divide. Whereas in humans, we have only 2 copies. So the chance of getting a mutated copy when you have only 2 copies versus when you have 20 would be a lot higher. If the cell passes that checkpoint and the cell is told everything is good to go, then the cell will start making something called cyclin. Cyclin is a protein. Cyclin is a protein that binds with another protein. So cyclin binds with CDK. So this is 2 proteins that come together. If you remember back to first semester when we did the mini tubers where we folded the piece of foam, we said that many protein complexes are made of multiple polypeptide chains. So this is kind of an example of that, where we have 2 proteins that come together to make essentially a mega protein. So we have cyclin that comes together with CDK to make a mega protein. That mega protein also has a little phosphate on it. And if you remember back to cellular respiration, ATP, a little molecule of ATP has 3 phosphates on it. And the way that that molecule of ATP gives the cell energy is by donating or giving a phosphate to a protein. And when the protein gets that phosphate, it causes a shape change that allows that protein to do a new job. So we then say it's like an energized protein. So my MPF here is an energized protein. It has a phosphate, it's made of cyclin, and it's made of CDK. And that MPF now can do a job that it couldn't do before. And the job that it can do is break apart the nucleus. During mitosis, DNA has to move around the cell. So the nucleus has to be broken in order for mitosis to occur. So MPF has to break apart that membrane around the nucleus so that mitosis can happen. You can see in the graph that the cell begins making cyclin during interphase, which is what we said on the previous slide. If we pass that G1 checkpoint, the cell is going to start making cyclin. As we see in the graph, it starts making cyclin during interphase once it passes that checkpoint. The cell also has this other protein called CDK. CDK is constantly at the same concentration. So CDK is there regardless of what the cell is doing. The amount of MPF fluctuates which is what's shown here, the amount of MPF fluctuates based on the concentration of cyclin. Because cyclin is one of the pieces needed to make MPF, you must have cyclin in order to have MPF. So as cyclin is getting made here, the amount of MPF can increase because it has like one of the ingredients needed. Then during mitosis, after the nucleus is broken, that cyclin is no longer needed, so the cell can degrade that cyclin. Well, when it's degrading the cyclin, it's degrading a piece of MPF, so the amount of MPF then goes down. So what this graph is showing is that the amount of CDK is constant, cyclin increases during interphase. When there is more cyclin, there can then be more MPF. And after the nucleus gets broken apart during prophase, cyclin is no longer needed, so it gets degraded during mitosis. There is more than just the one checkpoint that checks to make sure if there's food and space. So beyond that first checkpoint, there are 2 additional checkpoints. The next checkpoint is the G2 checkpoint. The G2 checkpoint occurs at the end of G2, which means that it occurs after S, because we have G1, S, and G2 is the order. So the G2 checkpoint will check to make sure that DNA was copied correctly. When the cell copies DNA, it's pretty darn accurate, but occasionally there are errors. They say about 3 in a billion errors. So 3 in a billion is like a really small percent. It's almost nothing. So it's pretty accurate. Most of the variations that we see when we talk about evolution are actually not caused by mutation. They're caused by sexual reproduction and rearrangement of traits, different traits in different combinations, more so than mutations. But that was a sidetrack. All right, G2. So the G2 checkpoint will check to make sure that the DNA was copied correctly. And if not, the cell has machinery to fix any errors that there might be. But there are occasionally mutations that get passed. So the G2 checkpoint is a small pause. You can see that these 2 checkpoints both occur during interphase, which also helps to explain why interphase is so long, because the cell pauses and makes sure everything is good to go before moving on to mitosis. Checkpoint number 3 is the M checkpoint. During the M checkpoint, the cell will make sure that the DNA is aligned properly at the center of the cell and that the DNA is all connected to the spindles. So if we imagine a cell in metaphase, I have the chromosomes lined up in the center. The spindle needs to pull those chromosomes. Some will get pulled that way, some will get pulled that way. Well, you better be sure that the DNA is hooked properly to the spindle. Otherwise, that chromosome, like this little X shape, would not get ripped in half. So then we'd end up with the 2 sister chromatids moving to the same side and the other side getting neither of the sister chromatids. Whereas what we should get is half of it moving one way and half of it moving the other way. So the M checkpoint makes sure that the DNA is all connected to the spindles. Signals for a cell to divide can come in a variety of forms. So one example is called a growth factor. A growth factor is a protein that stimulates cells to grow. So a growth factor is a protein that binds to a receptor in the membrane and then tells the cell, all right, let's go, time to divide. This diagram on the left is essentially saying that if you give cells growth factors, the cells will multiply much more than if the cell is given no growth factors. So a growth factor is one way that cells can talk to each other and identify when to divide. Density-dependent inhibition is another concept that has to do with stopping cell division. Density has to do with how crowded it is, and inhibition means to stop. Generally, cells will stop dividing when they become crowded due to a lack of space. That's what we saw with the G1 checkpoint. So shown here on the left is a diagram where there's cells in a dish. They replicate until the entire bottom of the dish is covered in cells. The scientist then removes a small patch of cells. Those neighboring cells that are left in the dish realize, oh, my neighbor is gone. So they replicate to fill in that missing hole. But generally, once they are next to their neighbor and all cuddled in tight, they're not going to keep replicating. However, cancer cells that ignore checkpoints will continue to divide, and they will grow three-dimensionally when there is no space left. So if left for an extended period of time, these cancer cells might spill out of the dish. They're growing, ignoring the fact they're running out of room, whereas normal cells would stop growing once the bottom of the dish is covered because they'd realize they've run out of room. Cancer cells tend to ignore normal cell signals, so they tend to ignore the fact that they are not being told to divide, and they tend to ignore those checkpoints. So that's then why we potentially might see a three-dimensional mass of cells like shown in this breast tissue, which we then call a tumor, cells that are ignoring the fact that they don't have signals to divide. Chemotherapy, of course, is one option for cancer treatment, depending on the type of cancer and person. But some chemotherapy drugs work by stopping the spindle from forming. So the spindle are those ropes that pull the chromosomes, and the chromosomes need to be pulled apart in order for division to happen. So the idea behind drugs that stop the spindle is you stop the spindle, you stop the division of the cell, you stop the growth of the cancer. There are other ways that anti-cancer drugs work, and you will research those in a paper.